And I want to give a special shout out and thank you to my son Jeff, who did the animations that you'll see today. Now, he's grown in to be a nice, generous, wonderful young man. And despite his generosity, though, I think he's kind of hinted to me that I might owe him a little something. So I've been kind of thinking back about all those years I changed his diaper, and I'm thinking, you don't want to go there, Jeff. We're going to start today in an unusual place. We're going to head out to the casino, because I imagine that most of you have heard it said before that if you sit down at a poker table and you don't see a fool, you're the fool. Well, that's me. If I don't play poker that often, but when I do, if I get an unlucky hand, what am I going to do? I fold, and I don't make any money. If I get a great hand, there's no poker face here, I get all excited, so everybody else folds, and I still don't make any money. But a good champion poker player can pull in a big pot with a bold bluff. Even if he's given a 7-2 offsuit to start Texas Hold'em, which are the two worst cards you can start with, because they're the lowest numbers you can get without being able to do a straight or a flush with subsequent cards yet to come. Now, he could get lucky, get triplets or full house. But a good poker player can use a bluff even if they don't get lucky, get everyone else to put in their chips, and then get them to fold. Throughout human history, one of the most unluckiest hands that people have been dealt is infections. I don't need to remind you how human populations have been ravaged by epidemics of polio, of yellow fever, smallpox, mumps, measles, rubella. But we've solved these problems. We've solved them by bluffing the immune system. Vaccines are a bluff. Vaccines are fake infections. They're not real, but they're close enough to be real that the immune system thinks they're real. It revs up, and it's ready to go when everything is, when you really need it. So that's been successful. Nowadays, one of the most serious and scary 7-2 offsuits that you can be dealt is the diagnosis of cancer. Cancer remains the number one cause of death due to disease in children. So you may have heard all the successes we've had with leukemia and some other cancers where we can cure 80-90% of patients. But there's a lot of other kinds of cancers, and overall, it still remains the number one cause of death in children due to disease. And overall, it's the number one cause of death for everyone, the number two cause of death for everyone behind heart disease. So it remains a serious problem. I'm going to tell you how I think we can trick the immune system into thinking cancer is an infection and make great strides in treatment. But first, let me bring it home to a little bit more of a patient story. I first met Katie when she was 8 years old, and her parents thought she had a cold. She couldn't breathe, head was stuffy, headaches, malaise. Unfortunately, the symptoms didn't go away, and so when we looked further into it, we found that her nasal passages had been filled up with a malignant tumor called rhabdomyosarcoma, and it extended into her brain. Now, we're left with just using the blunt instruments we have, which is surgery, chemotherapy, and radiation, to treat her. And those sometimes work, especially when it's not an advanced cancer and not spread. But they also cause a lot of side effects, both short-term and long-term effects that affect quality of life and organ function, etc. So we really need to do better. Now, in order to do better, we need to understand what we're up against. And there's been some really exciting findings about cancer biology, and I'm going to teach them to you. So at the end of this 10 minutes, you guys are going to all feel like, if I've done my job right, you're bona fide card-carrying cancer biologists. And because I'm going to put it in terms that we hopefully can all understand. So the three things I'm going to teach you are really come from Harry Potter. Bertie Bott's beans. Bertie Bott's every flavored beans are of all flavors, all types, just like cancer. The invisibility cloak, which is self-explanatory, and secret passages. If you can't read it there, if cancer could talk, I'm sure this is what it says. I solemnly swear I'm up to no good. So I think you all know the basics from high school biology about cancer. You start with a single cell, and eventually that cell acquires the ability to divide uncontrollably, And then those two cells divide and make more copies of themselves and more copies of themselves until you get this huge mass of cells that, say, fills up your nasal passage. One of those cells breaks away through the bloodstream or the lymphatic channels and goes to another organ. And over there, it also does the same thing, and that's the metastasis. But hold on, not so fast. Let's start over. When that cell divides, those two daughter cells really aren't the exact copy of the original one. They're more like copies of copies. And when those divide, the copies of copies of copies. So every cell is slightly different. It may be acquire genetic mutations, it may require differences in gene expression, but every time a cell divides, it's an experiment the cancer's trying to do. Can I make a better cancer? Can I grow faster? Can I spread better? Can I resist therapies? So this we call heterogeneity, and it's just like Bertie Bott's every flavor beans. And when one of those beans travels throughout the body to another organ and sets up shop, it does the same thing at the other site. So you get heterogeneity in both places. So that is one explanation why cancers are often resisting treatments. Now, some of these cells are so different from normal that the immune system ought to recognize them as foreign. So T cells, shown here in blue, that would come into the tumor, which they often do, and recognize it as foreign, they bounce off the tumor essentially. They can't see it. And we've not understood why for a long time. And we now know why, and it's what I'm showing here. Normal tumors actually recruit normal cells to become part of the tumor mass. These cells are macrophages, suppressor cells, suppressor T cells, myeloid-derived cells. These normal cells they don't succumb to chemotherapy and radiation. And what they do in the tumor is they help feed the tumor, they help it grow, they change its biology. They help it resist treatments. So we need to understand these invisibility cloak, which I've shown here in the sort of haze, the fog, because these cells are also produce substances in the microenvironment that suppress the immune system. So now we know that's why when T cells do recognize some of these different cancer cells as foreign and they invade the tumor, they can't do anything because of all this invisibility cloak that surrounds the Bertie Bott's beans. The final thing we've learned about cancer very recently, which is amazing and exciting and may explain why we can't have a hard time curing patients with metastases. Every place there's a tumor in the body, they exchange information. So tumors pass tumor cells to other tumors, and they pass some of these normal suppressor cells, and they exchange information. They protect each other. It's a network. They're communicating. They're supporting one another. So we have to treat them all at once because the others will just repopulate the original or acquire more resistance. So this is an amazing piece of biology, and thanks to my son, I think it's an amazing animation. So what we're doing in our lab is directly injecting tumors with viruses. And these are attenuated viruses. They're crippled. They don't really cause disease, but they're still potent enough that they can infect tumor cells and kill them directly, and they can simulate an infection. And what does the body do? It brings in a lot of T cells for an infection. So we're ramping up the T cell infiltration into tumors, and some of those T cells, yes, they're fighting the virus infection, but some of them are fighting the tumor as well. So we're really enhancing the ability of those cells to attack tumor. And that hasn't taken care of the invisibility cloak, so we're combining that virus treatment with drugs that are targeting those molecules that form the invisibility cloak, and that lays bare these tumor cells that are now able to be killed by other drugs, by virus infection, by the T cells themselves. And if we can do this systemically and hit all those sites at once and stop them from communicating with each other, which we're doing also by giving the virus intravenously, then we think we can make real strides. So just to recap, on the left we have traditional therapy. Tumors are protected. And again, I'm talking about mainly advanced cancers, some of the ones that haven't a lot of that heterogeneity, that maybe all one flavor, or that don't have much of an invisibility cloak, that are localized. We can treat those. We've learned that, but we just haven't learned yet how to attack a tumor like this that's spread, that communicates with each other, it's heterogeneous, has an invisibility cloak, and has secret passages. And we think with immunotherapy, like viral therapy, and actually other work going on here where we're trying to harness the immune system to attack tumors, that we can achieve tumor cures. Now, you may look at me and say, Dr. Cripe, you are crazy. This is phantasmagorical. You need to get a magic wand to do this. Go shop at Olive Anders in Diagon Alley. Well, we are making progress. So I wanted to explain where we're at in this timeline from idea to cure. So if you think about how we make progress, we usually start answering questions in the lab. Lots and lots of questions. Which tumor types, which viruses, etc. And eventually we bring those to the clinic. And we test it in people. And then we learn things in people that we then take back to the lab and try to answer questions in the lab. And this iterative process, create a new clinical trial. And eventually we learn as much as we need to affect as many cures as possible. So where are we on this timeline? Well, here at Nationwide Children's Hospital, I put us right about there. We've done a lot of work in the research laboratory. We've started clinical trials. We're learning from those, and we're advancing more clinical trials. And we're trying to do this for pediatric cancers. There's actually a company out there that already has an FDA-approved virus for the treatment of adult patients with melanoma. We're trying to learn to use those viruses in combinations, which viruses work good for which tumor types, etc. And just to show you an example, here's a patient, a CT scan and a PET scan combined together. CT shows the structures, PET shows the activity, the glucose utilization. I've outlined the tumor for you. This is a patient who was enrolled in one of our trials. We injected the herpes simplex virus, crippled version, that wasn't going to cause disease. And the next image you see, we detect that flare-up of cells coming into the tumor. I showed you in graphics with the blue cells. So we can detect that, and then later you can see that exact area where those blue cells came in, the tumor's dead. You don't see any signal. So I don't need a magic wand. This effect is real. If we can understand the Harry Potter nature of cancer and really understand what we're up against, I think that patients with advanced cancer in the not-too-distant future can live out their dreams. Just like Katie did, actually, because she was one of the lucky ones. She didn't need to bluff her cancer. And just last month, I got this letter, kind of chokes me up every time, announcing University of Cincinnati that Katie is a candidate for the degree of Bachelor of Social Work with a heartfelt note. Thank you for all you did for me at age 8 that has gotten me here today. For patients who aren't as lucky as Katie, if we can learn to bluff cancer like a professional poker player, we can rake in a big pot, saving lives. So I call your bluff, cancer. I'm all in. Thank you very much.